Welcome, everyone. My name is Michelle Galicia, and I'm a postgrad pre-medical student at Charles Drew University of Medicine and Science. Today, we will be talking about e-cigarettes and metabolic disease markers of adipose tissue. As a short background, we all have heard of e-cigarettes or vaping and how it has become popular within the adolescent group. This poses a higher risk of consumption among Hispanics and other U.S. minority groups. Usage of e-cigarettes has been previously linked to cardiometabolic disease as a result of lipolysis and increased free fatty acids. Previous studies have demonstrated that e-cigarettes induce an inflammatory response associated with increased serum free fatty acids and cardiac dysfunction. In this study, we will test how free fatty acid may lead to adipose tissue remodeling in ApoE knockout mice due to the inflammatory effects of nicotine. Now, why is this important? This is important because such a finding is relevant in determining a role of adipocyte tissue on e-cigarette-induced cardiomyopathy. In Figure 1, which is a working hypothetical model for the induction of cardiac dysfunction, notice how exposure of adipose tissue to e-cigarettes leads to increased lipolysis. This, with an augmented infiltration of macrophages and lymphocytes, leads to increased serum free fatty acid and increased adipokines. All of this results in cardiac dysfunction and atherosclerosis. For our design methods, we use a device for free-moving exposure chamber of e-cigarette aerosol in which we place 3 different groups of ApoE knockout mice under different vaping conditions for 12 weeks. The first one, we put e-cigarettes with 0% nicotine. The second one, we put e-cigarette with 2.4% nicotine. And the third one was saline only, which is our control. Then we performed immunohistochemical staining in which we got samples of murine adipose tissue and fixed them in paraformaldehyde, embedded these in paraffin, and sectioned them using a microtome. We then examined these samples using the F480 stain, Mason's trichome stain, and the Sirius red stain. In Figure 2, you can see a picture of the device for the free-moving exposure chamber of the e-cigarette aerosol used for mice. It is important to note that the F480 stain is used to localize filtration by macrophages, signifying an immune cell induction of inflammation. Here we see Figure 3A through C that it corresponds to the F480 stain. We only see a significant localization of infiltration by macrophages in the 2.4% e-cigarette treatment, or letter C, as indicated by the red arrows. In the second row, which corresponds to the Mason's trichome stain, there is an accumulation of the extracellular matrix and adipose tissue in the 2.4% e-cigarette treatment only, or Figure 3F. These accumulations are markers of fibrosis. The third row, which is the Sirius red stain, it shows prominent formation of collagen fibers in between adipocytes and tissue exposed to 2.4% e-cigarette treatment, also indicative of fibrosis. In Figure 3I, here you see red plaques or red thickened fibers. Those are the collagen fibers that have accumulated. As indicated by the immunohistochemical staining, we have seen how the 2.4% e-cigarette treatment all led to markers of inflammation such as infiltration of macrophages and initiation of fibrosis in adipose tissue. This tells us that nicotine e-cigarettes can lead to the presence of markers of metabolic disease in adipocytes. Such findings confirm pathways of inflammation that may contribute to cardiac dysfunction induced by e-cigarette. All of this can result in oxidative metabolic dysfunction of other target tissues like the heart or liver. Here are some acknowledgements. And for any questions, please email me at the address found at the bottom of this slide. Thank you.